Good morning, students. Today's topic is hemoglobin and competition number 6.12, and today's subtopic is various hemoglobinopathies, in which we are studying sickle cell hemoglobin or sickle cell anemia. First, we'll see what is hemoglobinopathies. Hemoglobinopathies means abnormal sequence, primary sequence of globin chain. Abnormal primary sequence of globin chain. Hundreds of such hemoglobinopathies have been discovered, but important hemoglobinopathies are hemoglobin S, C, D, E and M that we are going to discuss today. First is hemoglobin S. S stands for sickle cell hemoglobin. Of the hemoglobin variants or hemoglobinopathies, hemoglobin S constitutes the most common variety worldwide. In this case of hemoglobinopathies, in beta chain, normally at 6th position glutamic acid is present, and in hemoglobin S, this glutamic acid is replaced by valine. So there is only change in the single amino acid is seen in this case. Due to this single amino acid substitution, there is polymerization of hemoglobin molecule inside the RBCs. Polymerization means all hemoglobin molecules combined together, which causes distortion of RBC shape, which becomes sickle shape. Normally the shape of RBC is biconcave. So now here it becomes sickle shape. Now at molecular level, what happens is, in DNA of normal hemoglobin, the codon CTC is present, but in hemoglobin S, this codon CTC is converted into CAC. Here the T is replaced by A, due to which when there is a transcription of RNA, the codon GAG, which is normal, is converted into GUG, which is abnormal. Therefore, when this RNA is translated, then at 6th position GAG codes for glutamic acid and GUG codes for valine. So in this way, the valine is there in the beta globin chain due to mutation in the DNA. Now we will see the mechanism of sickling. Before going to see the mechanism of sickling, you should know the structures of various hemoglobin molecules. On the left side, I have shown you oxyhemoglobin A. 4 units, alpha, 2 beta, subunits are there. Now this is oxyhemoglobin A, means when hemoglobin binds with oxygen. When this hemoglobin A donates its oxygen to the tissue, then it is called as deoxyhemoglobin A. After delivery of oxygen to the tissue, you see there are concavities or notches present on the deoxyhemoglobin A, which appears on delivering of oxygen. Now next structure is of oxyhemoglobin S. If you compare oxyhemoglobin S and oxyhemoglobin A, then you find that on oxyhemoglobin S there are some protrusions, or also called as sticky patches, present on hemoglobin S. Now when this hemoglobin S becomes deoxyhemoglobin S, it appears notches or concavities similar to deoxyhemoglobin A. Now if you compare the structure between deoxyhemoglobin A and deoxyhemoglobin S, then you will find that on deoxyhemoglobin A only the concavities are present or notches are present, but in deoxyhemoglobin S you will find 2 notches or sticky patches and 2 concavities. Therefore, when deoxyhemoglobin molecules come close to each other, then the notches or protrusions of one deoxyhemoglobin enter into the concavity of other hemoglobin, a deoxyhemoglobin S molecule. So in this way, all the deoxyhemoglobin S molecule combine together, whereas normal deoxyhemoglobin A does not combines. When they combines, they form a polymer, and this polymer then mechanically disturb or change the shape of RBC from normal biconcave shape to sickle shape. The same thing is written here. The substitution of hydrophilic glutamic acid by hydrophobic valine in the beta chain causes a localized sticky patches on the surface of hemoglobin molecule. The deoxygenated hemoglobin S may be depicted with protrusion on one side and concavity on other side. That just we have studied, or just we have seen. So that many molecules of deoxyhemoglobin can adhere to each other and polymerizes. Remember, hemoglobin S can bind and transport oxygen normally. The sickling occurs only under deoxygenated state. And why this sickling occurs only in deoxygenated state? Because sticky patches or protrusions are already present on hemoglobin S, but the concavities or notches are present only in deoxyhemoglobin S state. Therefore, it favors polymerization. Now, if hemoglobin A and hemoglobin F is present along with hemoglobin S in deoxygenated state, then see what will happen. Hemoglobin A and hemoglobin B, F prevent sickling. How they prevent sickling? Here I have given example. On the lower side, I have shown you a polymer of hemoglobin S. On the left side, I have shown a deoxyhemoglobin A. Now this deoxyhemoglobin A does not have sticky patches or protrusions. It has concavity. So it get fit into the polymer on this side, but the next hemoglobin S cannot come and bind to deoxyhemoglobin A on that side. In this way, it stops the sickling. The sickled RBCs forms a plug in the capillaries, or they form a blockage in the small capillaries. Sometimes occlusion of major vessels or large vessels leads to infarction of organs, mainly spleen. Infarction means death due to loss of blood supply. As these RBCs get clogged or plugged inside the capillaries, blood supply is hampered, and due to lack of blood supply, some major organs may show infarction or death. Hemoglobin S can be presented in person in two different states. First is sickle cell trait. It is represented as hemoglobin AS, and second is sickle cell disease. It is represented as hemoglobin SS, hemoglobin SC, where one half of the hemoglobin is S and other half is C, and hemoglobin SD means half of the hemoglobin is S, half of the hemoglobin is hemoglobin D. So these are various types of hemoglobin sickle cell diseases. First we will see sickle cell trait, which is more common. It is a heterozygous condition. Here, out of two alleles, one allele is mutated and one allele of gene is normal. So normal allele of gene synthesize normal hemoglobin A, but the mutated allele shows synthesizes hemoglobin S. Therefore, in such person, two alleles are different. Hence, it is called as heterozygous condition. Now, in this heterozygous condition, 50% hemoglobin is normal, that is A, and 50% hemoglobin is S, which is sickle cell. Then, sickle cell trait as such does not show clinical symptoms, and such persons have normal lifespan. But when the persons of sickle cell trait exposes to hypoxia, due to some reason sickling occurs. For example, if such persons exposes to high altitude where oxygen tension is low, or if the patient of sickle cell trait also suffers from lung disorders, at that time these persons will show sickling, otherwise these persons are normal. Question: why the patient with sickle cell trait shows sickling of RBC only on hypoxia? To answer this question, you have to observe this figure. On the left side, I have shown normal situation. On the right side circle, I have given hypoxic situation. In the normal situation, see, a circle is there and I have drawn a vertical line. Left side of the vertical line is having hemoglobin A, which is 50%. On the right side of the vertical line, it is hemoglobin S, 50%. But we know under normal situation the hemoglobin saturation is 60%. That means only 40% of hemoglobin donates its oxygen to the tissue. That means only 40% of hemoglobin is in deoxygenated state. Now if 40% of the hemoglobin is in deoxygenated state in normal condition, that means 20% of hemoglobin A and 20% of hemoglobin S is in deoxygenated state. 20% of hemoglobin S only have protrusions or sticky patches, whereas remaining hemoglobin S, which is oxygenated, it does not have protrusions or sticky patches. Therefore, it does not participate in sickling. So, if you see complete hemoglobin, then you find that small part of hemoglobin S is deoxygenated under normal condition. Therefore, sticky patches are present only on small portion of hemoglobin S. Therefore, sickling does not seen. But if the same person is exposed to hypoxic situation, in case of hypoxia, hemoglobin delivers more oxygen to the tissue. Therefore, hemoglobin saturation decreases. The concentration of deoxyhemoglobin increases. The concentration of both deoxyhemoglobin A as well as deoxyhemoglobin S increases. When the concentration of deoxyhemoglobin S increases, it shows increase in the number of sticky patches or protrusions. Whenever there is development of sticky patches on hemoglobin S, the sickling occurs. So this is the answer why sickling occurs in hypoxic state only. It is said that hemoglobin S gives protection against malaria. It is observed that the high incidence of sickle cell gene in population coincides with area endemic for malaria. Endemic means a disease which is confined to some area. So wherever there is malaria cases are more, such patients have sickle cell hemoglobin. This hemoglobin S affords or gives protection against Plasmodium falciparum infection. Hence, abnormal gene was found to offer biological advantage to such persons. Sickle cell trait is very common in areas where endemicity of malaria is seen, like Central and West Africa, East and Central part of India, tribals all over the India. But due to slave tradition in the past, generally the slaves or the working people are deployed for working in the developed countries like Europe and America, and they take the slaves from African countries to their countries for work, and this due to this slave tradition there is spread of this gene from Africa to different parts of America and Europe. Now, how this sickle cell trait protect against malaria? Plasmodium the infectious agent of malaria completes its life cycle in RBCs. Sickle cell RBCs get destroyed early and hence their life cycle is not completed. In this way they protect from malaria. Now next is diagnosis of sickle cell hemoglobin. It is generally done by electrophoresis carried out at pH 8.6. At pH 8.6 proteins have negative charge. So here I have shown you 3 circles. In that 3 circles, these are nothing but these are 3 wells on electrophoretic plate. In the first plate I have applied a sample of hemoglobin A. In the second or in the middle well I have applied the sample of hemoglobin SS. Hemoglobin SS means hemoglobin sickle cell disease. And in the third well I have applied a sample of hemoglobin AS, means hemoglobin sickle cell trait, right? Then when I have connected this plate to the positive and negative terminal is on left side, positive terminal is on the right side, then I have seen that hemoglobin A have migrated very fast and 2 bands of hemoglobin A are seen at the pole where positive charge is present. Why hemoglobin A have migrated faster? Because in hemoglobin A glutamic acid is present, which is acidic amino acid. It ionizes and get negative charge at pH 8.6. Therefore, this due to presence of more charge on hemoglobin A, it migrates faster. Next, see third slide of sickle cell trait. Here I have again seen 2 bands. One band have migrated faster, equivalent to hemoglobin A, but other band have migrated slower. The band which has migrated faster is of normal hemoglobin A, and the band which have not migrated faster is of band of hemoglobin S. This is because just we have studied in hemoglobin S glutamic acid is replaced by valine. Valine is a neutral amino acid. Therefore, hemoglobin S has less charge as compared to hemoglobin A. Therefore, in electrophoresis it migrates at slow rate. In patient of hemoglobin S disease or sickle cell disease, 2 bands are seen again, but both the bands have slow mobility or they does not migrate as fast as hemoglobin A, and 2 bands are separated in the electrophoretic slide nearer to the point of application. Here the question, why hemoglobin S is slow moving band? Just now I have told you hemoglobin S has less negative charge as compared to hemoglobin A, because in hemoglobin A glutamic acid is present which has negative charge. In hemoglobin S valine is present which has no charge. Therefore hemoglobin A migrate faster and hemoglobin S is slow moving band. The same thing I have written here at pH 8.6 carboxyl group of glutamic acid is in hemoglobin A is negatively charged. Lack of this charge on valine of hemoglobin S make it less negative charge hemoglobin and decreases its electrophoretic motility towards the positive pole. So hemoglobin S shows slow moving band than hemoglobin A. In sickle cell trait, both bands of hemoglobin A and hemoglobin S are noticed, just we have studied. One more diagnostic test is available for diagnosis of hemoglobin S, and that is sickling test. Here we have to take a glass slide, then 1 drop of blood is taken on that glass slide. A reducing agent sodium dithionate is added to that blood and we have to apply a cover slip. We have to keep it for some time. After some time we have to observe this glass slide under microscope. Now the function of this sodium dithionate is that sodium dithionate convert the oxyhemoglobin into deoxyhemoglobin, and all of us know that if deoxyhemoglobin molecules are present in hemoglobin S, they get polymerized and sickling of RBCs is observed under microscope after some time. So these are the different types of sickling RBCs are seen under microscope. This is also a diagnostic test for sickling. Next is sickle cell disease. It is represented at hemoglobin S. Here two alleles are there and both alleles get mutated. So both alleles will synthesize abnormal hemoglobin S. Therefore, total hemoglobin is called as hemoglobin SS. They are inherited as recessive or codominant trait. One beta chain is inherited from each parent. If both parents are heterozygous for hemoglobin S, heterozygous means half 50% is normal and 50% is abnormal, then there is 50% chances of child having hemoglobin S trait, 20% of chances that child may have normal hemoglobin, and 25% of chances of having hemoglobin SS genotype or sickle cell disease. Sickle cell disease, if the genotype is SS, all hemoglobin molecules produced are abnormal and hence disease is manifested, and death usually occurs in the second decade of life. How will you manage this sickle cell disease? You have to give repeated blood transfusion to the patient for anemia, but due to repeated blood transfusion there is iron overload in the body. This iron may get deposited in the liver, leading to cirrhosis of liver. The treatment with anti-sickling agent like hydroxyurea, cyanide and aspirin may be given. Sodium butyrate will also induce hemoglobin F production with some clinical improvement. Now these are some questions and these are the answers. Now if one parent is heterozygous for hemoglobin AS, means sickle cell trait, and another parent is hemoglobin AC or AD, then child may have heterozygous traits like hemoglobin SC or hemoglobin SD. After seeing hemoglobin S, next is hemoglobin E. It is second most prevalent hemoglobin. In this case, at 26th position of beta chain, glutamic acid is present, which is replaced by lysine. It is primarily seen in orientals of Southeast Asia, mainly Thailand, Myanmar, Bangladesh and West Bengal in India. Heterozygous state means one allele is normal, which synthesizes 50% hemoglobin A. Another allele is mutated, which synthesizes 50% hemoglobin E. Therefore, heterozygous state is represented as hemoglobin AE, but this state is completely asymptomatic. Homozygous state means when both the alleles of gene are mutated, both alleles will synthesize hemoglobin E and the patient is represented as hemoglobin EE, which shows mild hemolytic anemia. At alkaline pH, hemoglobin E shows less motility as compared to hemoglobin A. Now hemoglobin C, here at 6th position, amino acid glutamic acid is present in beta chain, which is replaced by lysine in hemoglobin C. This hemoglobin C is mainly seen in black races. AC means hemoglobin AC is heterozygous, which do not show any clinical manifestations, but those who are double heterozygous means hemoglobin S and hemoglobin C, both the alleles are abnormal. One synthesizes hemoglobin S, one allele synthesizes hemoglobin C. Such are called represented as hemoglobin SC. They have moderate disease. Homozygous means both the alleles are mutated and both will synthesize hemoglobin C. They are represented as hemoglobin CC. They show mild to moderate hemolytic anemia. Again, hemoglobin C is slow moving band on electrophoresis. Next is hemoglobin D. It does not produce sickling. Hemoglobin D or Punjab results from replacement of beta chain amino acid. At 121 position, glutamic acid is present, which is replaced by glutamine. Hemoglobin SD is a severe condition. Here SD means one hemoglobin is means half of the hemoglobin is hemoglobin S and half of the hemoglobin is hemoglobin D. Such condition is represented as hemoglobin SD. It is a severe condition. Hemoglobin D Punjab is the commonest hemoglobin variant found in Punjabi population. Hemoglobin D migrates similar to hemoglobin S on electrophoresis. Therefore, diagnosis of hemoglobin D can be done by sickling test. Hemoglobin M, here just now I have explained, it is called as hemoglobin M, or it is not methemoglobin. Here either proximal histidine or distal histidine is replaced by tyrosine. To understand hemoglobin M, you should know this structure. This is a structure of, this is a structure of one hemoglobin subunit. Here if you see, iron is having 6 attachments. So iron is hexavalent here. 4 bonds form with 4 pyrrole rings. 5th bond of iron is with the proximal histidine and 6th bond is with distal histidine through oxygen. Now if proximal or distal histidine is replaced by tyrosine, iron cannot form a bond with tyrosine. In this situation, iron can form only 5 bonds and iron becomes pentavalent. When iron becomes pentavalent, its ferrous form is converted into ferric form, which we call as a methemoglobin. So methemoglobin does not binds with oxygen and it will leads to cyanosis. When in alpha chain, when distal histidine at 50th position is replaced by tyrosine, then it is called as hemoglobin M Boston. If proximal histidine of beta chain at 92nd position is replaced by tyrosine, then it is called as hemoglobin M Hyde Park. So these are about the hemoglobinopathies. Thank you very much.